Hi, everybody. My name is Joey Blankenship, and I'm an assistant professor here at the University of Arizona in Tucson. And we'll be representing one of the projects from the dry side of soil health. After I provide a brief opening with some context, I'll be passing things over to postdoc Dr. Kirsten Ball, master's student Chance Muscarella, and although he won't be speaking today, Samuel Rathke is my lab manager who is really one of the engines keeping this project going. So if you're unaware, dry land or arid soils are often on the cusp of becoming unhealthy or sick. On the physical side of soil health, these soils often lack physical structure or aggregation. Water infiltration tends to be too fast or too slow. Water in general tends to be scarce because of the dry climate and frequent droughts. There's often high wind and water erosion that harms the precious topsoil that is there. And then there's often fast photodegradation due to abundant sunlight, which causes decomposition of organic matter. On the chemical side of soil health, there's high pH, low organic matter, high carbonates, low nitrogen and phosphorus, and high salinity and sodicity. And finally, on the biological side of soil health, arid soils tend to have lower overall biodiversity. They tend to have lower primary production, greater vulnerability to disturbance, and microbes are more likely to outcompete plants for nutrients and other limiting resources. Soil health research and outcomes most desired by desert ranchers and farmers include revegetation for ranchers. So there's a lot of areas that have become barren and just are devoid of vegetation. There's high rates of erosion, so there's a desire for less of that water and wind erosion. Desert farmers are craving overall soil health conservation, primarily building organic matter, as shown from results here from a survey of Yuma leafy green farmers. They're desiring reduced inputs of water, fertilizer, and biocides. Obviously, water is a big one in the desert. And also strategies that either decrease or at least do not increase pH and salinity. And we're finding some counterintuitive patterns in the desert that we can't necessarily assume that diagnosing and treating soil health is the same as wetter climates. For example, microbial species and biofertilizers may not be able to survive the intense desert heat, so we might need different sorts of strategies and to use different sorts of microbes. Organic amendments do not always increase or improve soil structure. We've had a site where we added large amounts of compost and biochar and actually found a decrease in soil aggregation. And finally, we also can't necessarily assume that cover crops always improve soil health and sustainability, because in the desert, adding more irrigation actually might increase salinity and water use. So now I'll pass things off to Kirsten to hit on more of our specific challenges.
So as Joey mentioned, we are starting to see some counterintuitive patterns with some of our field trials, and it led us to a question about whether or not we're actually looking in the right place when it comes to quantifying arid system soil health. And that also led us to ask questions about our current and proposed metrics. So are they going to be suitable for measuring soil health in arid systems? And if they're not, what can we do about it? For example, substrate-induced respiration, one of our DSP's metrics, has been shown to actually severely underestimate soil microbial biomass in arid alkaline soils. Soil organic matter, of course, critically important for soil health because it maintains so many different soil health functions. But in our arid systems, we actually tend to have, in a lot of places, more inorganic carbon versus organic carbon. Now, that is something that we are measuring, as most of you would know, by a derivation alone from our organic carbon, but it's not something that we tend to be focusing on as one of our metrics for predicting the impacts of management. And then you're going to hear Chance talk a little bit about soil enzymes and how applicable they are for arid systems and what some of the pitfalls might actually be for their use. So we have a bunch of different systems that we are looking at within our DSP's program. And one of those is examining soil organic carbon across an agricultural chronosequence. So we have a native reference condition, and we're comparing across 3 different types of tillage. A conventional tillage where all of these fields are all alfalfa fields, by the way, so they're all being cropped in exactly the same way. The soil types are fairly similar. There's some differences in them, but we've been able to account for those differences within our models as well. So our tillage treatments are conventional tillage for more than 10 years, minimal tillage for the last 3 years, and minimal tillage for more than 10 years. So we have a really good set of conditions which we can compare across. So we ask questions about whether or not reduced tillage actually increases soil organic carbon in these carbonate-rich soils. And we actually found some really interesting things. And the reason that we found these interesting things was because we were looking at our data slightly differently. We were accounting for that inorganic carbon component, as is so important in our arid systems. So I just wanted to share a little bit of data with you. So if just looking at the tillage treatments, for brevity, you have your soil organic carbon to inorganic carbon ratio on the x-axis, and these have all been mean-centered. So I think the mean of this soil organic to inorganic is around 2 in across this whole data set, and you're looking at 1 absolute point increase and decrease above and below the mean through here. So you can specifically see that in our conventional tillage, where we would potentially expect there to be less organic carbon, because the tillage is actually liberating the carbon from the soil, we're actually getting increases in soil organic carbon as the SOC-IC ratio increases. When in the minimal tillage treatment, especially the 3 years, there's really no relationship at all. And in that 10-year minimal tillage treatment, you still have a significantly higher amount of soil organic carbon at that mean SOC-IC ratio. So this kind of indicates to us that we need to look at our data in a slightly different way if we really want to tease out the interesting parts of what's happening in these arid systems. So I'm going to pass over to Chance now, and he's going to talk to you about soil enzymes.
So let's talk about what exoenzymes are and what their role in the soil is. Exoenzymes are enzymes that are released by microbes to decompose organic matter in the soil. They're how microorganisms break down materials from dead plants and other microbes. They're important mediators in the transformation of nutrients in the soil. Nutrients like nitrogen and phosphorus go through many cycles as they're used up by the plants and microbes that are in the soil, changing them from one form to another. These enzymes convert the nutrients to a usable form by releasing them from an insoluble form that's attached to an organic substrate and pulling off the nutrient into an inorganic soluble form. An example is phosphatase, which pulls a phosphate off an organic compound and turns it into the inorganic form of phosphate that is soluble and available to the plants. So, can enzymes be used to assess soil health? Exoenzyme activities, or EEA, can provide some insight into the potential level of nutrient transformation in the soil. Combined with data on other dynamic properties, such as plant-available nutrient concentrations or organic matter content, exoenzyme activity levels have the potential to be a useful indicator of the functional potential of the soil in its current condition. On paper, EEA meets 3 of the 4 requirements of a useful soil health indicator. Lehman et al. Have stated that a useful soil health metric should be relevant to soil health and ecosystem functions, which they are due to their role in transformation of nutrients, be sensitive to change, be practical, and be informative for management. As it stands today, it's not clear whether enzyme activity measurements are capable of informing management processes, especially in arid soils where these soil health metrics have been less well studied compared to wetter, more temperate systems. What are some potential roadblocks for incorporating EEA as a soil health indicator in arid soils? We need to ask, do enzymes play the same role in water-limited systems as they do in wetter systems? Enzyme activity is often positively correlated with organic matter content, nutrient concentrations, and pH. Arid soils tend to have lower organic matter content and are often lower in available nutrient levels while having alkaline soils. Here in southern Arizona, our soils often range from pH of 7.5 to 8 or even higher. How will enzyme activities be affected by the high pH but low organic matter concentrations that we have? Will we still see similar trends to those found in more temperate regions, or will we see substantially different trends? These questions make it clear that we need to improve our understanding of how exoenzymes respond to arid conditions if we're to be successful in applying them to assess soil health in the desert. To achieve this deeper understanding, we're also comparing dynamic soil properties between unmanaged native reference conditions to soils from 6 vineyards in southern Arizona with a variety of management practices such as a history of reduced tillage and cover cropping trials. Comparing the trends in enzyme activities between the managed soils and reference conditions, combined with other soil health metrics like organic matter and plant available nutrient concentrations, will allow us to tease out the quantitative relationship between EEA and other dynamic soil properties. And now I'll hand it off to Kirsten to zoom out a bit and wrap us up.
So what we're really proposing is a new framework for soil health assessment in arid systems. The first component of that, which we spoke about initially, was understanding a little bit more about our system. There remains a heap that we don't know, that we don't even know. And our theories about arid system soil health need constant updating. And so that's a very large part of the project, is to update that understanding and then to translate that understanding into improving our measurements. We're asking questions about whether they're effective, and if they are not effective or potentially ineffective, how are they limited? How does the theories of arid system soil health inform our ability to improve our measurements? And if they can't be improved, then what can we potentially replace them with? And our measurements, of course, feed into our models in helping us to properly understand and describe what's going on in these systems. You know, how does our data that we're deriving from our systems actually support the theories that we are developing? What are the implications for management? You know, how do we help our agricultural managers to better manage their soils on the basis of the theories that we've developed and the measurements that are helping us to quantify what's actually going on? So I want to thank you all very much for listening to our talk, and we look forward to having some of you reach out to us and have a chat about arid soil health. Thanks.